Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last class we started our hands on and we focused on installing R and setting up the system. Okay, so we will continue that in this class, right. So, we will set up the system in R. I will show you how you can get this data and how you can start loading packages in R for all sorts of analysis that will come afterwards. Just to remind you, the goal is to perform a differential gene expression analysis and to identify genes that show differences in expression between two different conditions. So, I have also showed you the data set where we have 16 samples and we have raw count data for about 6,800 genes. Okay. And in the last class we loaded that data in R, right, and we were analyzing or looking at some of the statistics and how to access some of the elements in the data. Okay. So, we will continue that, we will go into much more advanced analysis now and we will also see how to load packages. Okay. So, this is what is the agenda for this class. Now, let us move on to the R part. Okay. Okay. So, we have the R installed, right. So, here it is, we have the data, we have loaded the data and this is the data we have. Okay. So, head data, this is sorry, head tab, right. So, the name of the variable is tab. So, we have the data here and I have showed you how we can access the data. Now, with R we can do a lot of very interesting things. We can actually also look at the data, we can plot the data, we can do statistical analysis, right. We can do a lot of things. Okay. So, one of the first things we can just play around a little bit, so that you get a feel for the data and how to handle the data. Okay. Of course, I also encourage you to play around, right, only then you will learn a lot more about R. Okay. Not just by reading the instructions or manuals, because sometimes those might be very difficult to understand unless you try yourself in this console. Okay. Okay, so, what I will do, we can simply generate for example, here we can probably generate something like an histogram, okay, by accessing this histogram tab $S1. Okay. So, I showed you, right, this is a very convenient way of accessing the column data in R, right. So, let us say we want to create an histogram and we want to see, right, what is the distribution of this read count in the sample S1. Okay. So, for that we are doing this histogram tab $S1, right, and as you can see, right, the histogram is, it looks a bit weird, right, because we have this frequency here and then most of the values are within 50,000 and then you have probably very few values around above 50,000, right. So, what you can do is we can now change this, instead of frequency histogram we can make it a density, right, probability equals to true, because for each sample the number of counts or the frequency will be different, right. So, we can make it density. So, we can say probability equals to true. We can also change the x axis limit, right. So, instead of this 0 to really big limit, we can simply say x lim equals to c 0 to 50,000, right. So, we want to see up to 50,000 because rest of them would be outlier. Now, this is a problem, right. The histogram is this, like you see just one bar here, right. Again we can solve this issue okay, by using this command called breaks. Okay. So, this will decide, right, this will actually design these breaks in such a way, right, we can see these bars, right, different heights. Okay. And similarly we can increase this, right. So, maybe we can make it 500 and now you see probably a bit nicer, right, it to look at, and we can also change the limit further, right, and you can see this, this is how the distribution looks like. Okay. So, this is something that you can do very easily, and one thing you will probably realize as you start using R is that it is very flexible and you can actually add a lot of things in your data. Okay. So, one of the things maybe we want to see, let us say we want to draw a line at let us say 100, right. So, where the count is 100, right, and we want to see like what fraction of these genes actually have read count more than 100. Okay. So, for that what we want to do here is that we want to add a vertical line at 100 here. Okay. So, in R we can do that very easily. Line, since this is a vertical line, we can say V equals to 100, right, and we can then give also a color to that line, so that we can clearly see this line clear, right. We can also mention line width, right. So, if we want a thicker line, we will increase this width, and also line type. So, LDY is line type. So, maybe we wanted a dotted line, right, or a dashed line. Okay. So, again you can change that with this LTI command, and you can see here, right, there is a line that appears around here. Okay. This is the blue line. Maybe we can change it to 1000. So, this would be more visible. Okay. And you can see there is another line. Okay. So, this is something that is really possible in R, right. You can add these vertical lines, also the horizontal lines, using these commands after you have drawn a plot. Maybe let us say you want to change the labels of this histogram, right. So, how do you change these labels? Is something like X lab, right, and X lab is the, we will say, raw count, right. This is the raw count. X label is raw count, Y label is fine for us, and main title is also fine. So, you can see now the this label has changed to raw count. Okay. Instead of this something else, it is now raw count. Okay. So, this flexibility is there, and we will actually explore this a bit more when we actually go for the visualization part, right. So, we have this result visualization part for DSA analysis, and we will explore that when we go there. Okay. So, another thing we can do, maybe we can want to see, right, how good these counts are, right, how good this correlation between counts between these, let us say, S1 and S5 samples are. Okay. Maybe can we see that visually? Okay. So, I will tell you, right, this information will come later. So, S1 and S5, these are technical replicates of the same sample. Okay. So, they come from the same condition, these are simply technical replicates. Okay. So, can we see how good this correlation is, right, between the gene counts, right, the raw counts that we have for the genes. Okay. You can do that, right. You can simply say plot, we are getting the column data now, right, and we can access these two column data. So, what we are doing simply is we are saying plot x and y. So, x is that this column data from the S1 sample, and y is the column data from the S5 sample. Okay. And you can see, right, this is how it looks like. Okay. Maybe this is not clear, okay, because there is one extreme count, right, very high count for one gene here that you can see, right, and rest of them are concentrated here, all these 6800 genes are concentrated around this region. So, that is why they are not really visible. So, what you can do is we can plot this in log scale, right, and in R you can do that very easily by saying, because we are dealing with positive numbers only, right, so, or 0. So, we can simply say log x y. So, it will transform both x and y axis into log scale. Okay. And this is something what you see now, right. So, you see there is pretty good correlation. Okay. And one of the things you can probably see here, it says 273 x values omitted because they are less than equal to 0, because they are probably 0, there is no count. So, anyway, we are not really that much interested where we have this 0 counts, and the rest of them are here. Okay. Now, what you can do also, so one thing you probably notice from this analysis immediately is that the reads or the genes that show very low count, there is a lot more variability, right, between these technical replicates. You can see this part is quite diffuse, and the difference between two replicates is large. Okay. This is what we observe in RNA-seq data always, right. So, the reads or the genes that show low read count or are lowly expressed, they show more variability. Okay. This is expected, but then also there are some genes where we see some variability. Okay. And this is something, okay, that is part of our experiment. We have this technical variation that we will have to deal with. Okay. So, what you can do, we can probably now also, this part looks very dense, right. So, maybe we can change it a little bit. We can also change the colors, right. So, there is all these options that you can add here after we have said plot, right. So, you can say we want these filled circles, not the open circles. We want to change the colors. Okay. Now, instead of using this blue red color in R, you can also use this RGB scale. Okay. And you can specify which color we want. Okay. Let us say we want blue color, but a very light blue color. Okay. So, the first number here, so in here you will have 4 numbers. Okay. When you say RGB, the first number is for the red scale. Okay. We are saying 0 because we do not want red. The second one is for green, right, and the third one is for blue. We want blue points, so we are using this. If you want pink or some combination of red, green, blue, you just mention these numbers, right, and different fractions, and you will get these different colors. And the last number is for transparency. Okay, so we want this only want these points to be only 50% transparent, because we want to understand the density of these points inside, right. Otherwise, they become fully black in here. Okay, so all these things we can do, and we can simply write this. You can see, right, this has changed now. Okay, the color these types have changed, right, and this color has also changed. Maybe we want even lighter colors, right. So, we do not want this darker color. So, we change the color code. So, from 0.5 to we reduce it to 0.2, we also increase the transparency, right, and this becomes a bit darker now. Okay, so you can see this now getting a bit more transparent, but there are so many points around here that we still have a very dark region. Okay. Now, maybe we want to see this correlation better, right. So, maybe we can draw a y equals to x line, okay, and maybe we can add as a dotted line to this plot. Okay. So, before we go there, maybe we want to change the x axis levels, right. So, you can say x lab is S1, right, y lab is S5, right, y label. We can change the size of these levels, we can increase them, C x dot lab 1.4. So, any number above 1 would increase the size, any number below 1 will decrease the size. Okay. And by the way, you can do the same thing for axis as well as for the points that are in the plot. Okay. So, we will not touch the axis and the points at this point at this moment, we will do that later on. Okay. Okay. So, as you can see, S1 and S5 levels have appeared now, right, in the x axis level and the y axis level, and now we want to add this line y equals to x. Okay. So, that will actually tell us how good the correlation is. Okay. So, for doing that, line a equals to 0, right, b equals to 1. So, these two numbers, the first number is the intercept. Okay. So, we want this line to pass through 0 0. Okay. So, we are fitting this y equals to bx plus a, right. So, or a plus bx, right. So, a is the intercept and b is the slope. Okay. So, b equals to 1 mean the slope is 1, right, and then we can say the line width, right, line type 2. So, this would give us a dotted line, and we can perhaps say the color would be red here. Okay. We want a red color because we will get a good contrast perhaps. Okay. And you can see this y equals to x line here, right, and you can see most of these points are falling around this, or very close to this y equals to x line, except this group of points for these genes that show low read count. Okay. So, this is again expected, we have talked about this because there are a lot more fluctuations that will be observed in this data. Now, this is all good, right, we have generated a very nice plot. So, one of the things now we want to do, we want to actually look at the correlation. Okay. Can we do this correlation calculation, right, and it turns out you can in R, and the command that we use. So, let us say value equals to, right. So, we are assigning this value here, correlation dot test. Okay. So, this is the command in R, okay, and we just need to give the names or the point to the data sets for which we want to calculate this correlation. Okay. So, it is very simple, code dot test tab $s1 tab $s5, right, these two data sets or these two columns we want to calculate correlation. Okay, and this correlation is now generated and stored in this variable value. Okay, and we can then type value, and it says okay, Pearson's product moment correlation. So, it is Pearson's correlation, and then p value is less than a certain threshold. You can see this is 2.2 into 10 to the power minus 16, that is actually the limit, and the correlation value here is 0.98. Okay. So, this is very good correlation between two replicates, right, and you can also get the exact p value. You can simply write $p dot value. Okay. So, again like the data set for this variable, you can also have this dollar, and many of these values are, you can simply find out by typing this, using this tab after you type the dollar. Okay. So, the tab dollar statistic will give you that is statistic that has been generated here. You can see this, right, 80.85. Similarly, dollar p dot value, right, this will give you the actual p value. So, it is here 0. So, it is a very extremely low p value, right. So, it is not 0, of course, it is extremely low p value, because these two data sets are very highly correlated. Okay. So, you can change this correlation format again, right, and you can say method equals to, so instead of Pearson, we let us say want this Spearman. Okay. So, it is giving me an error because I have typed in capital, S should be in small. So, it will tell you if in case there is an error in your command, it will tell you okay. So, argument should be one of Pearson, Kendall or Spearman. Pearson is the default correlation it calculates. So, if you want the Spearman correlation, this is the rank correlation, right, it will give you some values. It also gives you warning, right. So, because you probably have these ties in the data, right. So, again it will not give you the exact one, but it is okay, right. Again this p value you can get, you can simply write value, it is 0.983. So, it is slightly different from what we got for Pearson's. Okay. So, for this data it does not really matter whether you are taking Pearson's correlation or Spearman correlation, but for some data sets and also depending on the situation, you might want to choose the right one, right correlation that you will use. Okay. So, one of the things now I wanted to show you, right, we have seen, right, we can now plot these data sets, we can look at correlation, we can do very nice generate very nice plots, etc., using R. So, what I will do now, I will actually show you where you can get these data sets, right. So, if you have your own data, you can generate this raw count data using this mapping and then counting, right. But if you do not have your own data, if you want to do this hands on, right, you can of course get access to some of the data sets that are freely available. Okay. And these data sets are available in NCBI Geo, right. I have shown you before, I think, but now I will show you where you get these actual data sets. Okay. So, what I will do is I will go to this series part, right. So, if you search NCBI Geo, it will come here, and once you go into the series part, you can see a lot of data sets that are coming in, and you can see these very recent data sets that have been just deposited here. Okay. We can take any data set, there are like huge number of data, but you can see this expression profiling by high throughput sequencing. Maybe I will zoom in a bit, right. You can see these different studies that are coming in, you have the expression profiling by high throughput sequencing, etc., right, and we can go on, right, and you can download anything you like, any data or any data that is relevant for your work, right, from this data set, right. And let us say I click this one, let us say this is a human data set. Okay. What happened, I do not know. So, maybe I will click here. Oh yeah, sorry. So, I'll because when I click on that, actually it is going only to the human data sets, I think. So, I will just click on this GSC number. So, every study will have a unique GSC number, and once you click on that, you will get a lot more detail about the data. Okay. So, you have this title, you have the organism, expression profiling by high throughput sequencing, right, and then of course, you have the sample types, right. So, you have wild types, 3 replicates probably, and then 3 knockouts. Of course, we need to look at the paper, etc., and see the experimental design carefully what is actually happening. Okay. And here you can find this GSC 23552 gene expression dot xls.gz, right. So, this is actually this is I think is raw count data or maybe normalized count. This will be actually described whether this is raw count or normalized count data, but some sort of count data that is given in an XLS file. Okay. We can also look at other data. So, where we can actually get this text file, right, and it will mention whether it is a raw count. So, again now this is another study, right, and you can now see here this is where all these files would be provided. This one is a normalized count data, right. So, this is, you can see this FPKM normalization has been done already, right. So, that is why it says FPKM expression genes dot text, right. So, this is a normalized count data, we know straight away. Okay. So, depending on the type of analysis that you want to do, right, what I will suggest, you probably should try to download raw count data, because we will go through the differential expression analysis. Okay. And when you want to go through the differential expression analysis, we will use the DSEC2 platform, which only takes raw count data, right. It does its own normalization, etc., and it will do its own normalization before it does this differential expression analysis. Here is another study again, and you can see this fpkm.txt.gz. So, this is again FPKM normalized. Okay. So, this is something you can look at, right, and carefully choose the right data for your analysis. And of course, you do not want to choose a really big data, but a small data set that you can run in a reasonable amount of time. Okay. So, hopefully, this is clear. Now, we can actually go back to R again, right, and we can see how we can install some of these packages. Okay. All right. So, this is all fine. Now, we want to use some packages, right. So, as we have mentioned, we want to, let us say, use this DSEC2 package, right, and for plotting or for different types of tests, we want to install certain packages. Okay. So, if you go back to this R home page, right, on the left side, you will see this packages option. Okay. And for in this packages option, you can now go into table of available packages, right, that are available for this system, for this version, etc., right, and you can see very new packages that are coming in. Okay. And these are the names of these packages that are available, and very short description on the title of the package, right, what it does. Okay. And you can find a huge number of packages, right, and only some of them are required for our purpose, right, but then again we can install them through the terminal. Okay. You do not have to go through this, right, and find out, right, which package is doing what, right. If we know which package we want, we can simply install it through the R console. Okay. So, one thing I will do, maybe what I will try now is I will look for a package that we can install. Okay. Okay. So, let us see, we need a package something like, let us say, this ggblend, right. So, here it will again once you click on that package, you will come to a page like this, and it will have a description of what this package does, right, and which version of R you can use for this, right. So, this is mentioned here, and again you have some more details about the authors, right, and also the reference manual here. Okay. So, this is the most important document. Once you click on that, you will have a PDF manual that will describe the functions that are available within that package, what these functions do, along with some examples. Okay. So, this kind of information would be available for all packages. Now, let us say if we want to install this package, right. So, let us say ggblend. Okay. We want to install this package here. Okay. So, to do that, we can simply go to the R console here, okay, and we will clear this window. So, that will be easier for you to see. Here you simply write install dot packages ggblend. Okay. Be careful of this with the spelling as well as the capital letters. So, you can simply write this install dot packages ggblend. You can choose any mirror, we will just choose the default one, the 0 cloud, right, and it should install this in a moment. Okay. So, you will see something like this, which will just install this. So, here you see like this is installed. In some cases it might install some of the dependencies, etc., and some of the required packages also along with this package. Okay. This process is exactly similar if you are also working in the Linux. You can simply type install.packages ggblend. Okay. So, now if you want to use this package, what you have to do is you have to load this package by saying library ggplane. Okay. So, now it is saying okay, this package was built under R version 4.3.1, but I have 4.3.0. So, it might give you some warning message, but that is okay. So, you can also use certain packages that will use, right, for example, ggplot2. Okay. So, whatever package you want to use, you simply type, if it is installed, you can simply type library then package name, and this will be loaded, and you can now use these commands from this package. Okay. If it is not installed, it will tell you okay, cannot find this package, the name of the package, right, and then you can say install.packages and the name of the package. Okay. So, this works for most of the R packages, right, but there are other ones as well. Okay. So, when we are discussing about the theory etcetera, about bias correction, different methods etcetera, right, we talked about that there are some bioconductor packages, okay, and in R bioconductor. Okay. So, this is actually a part of R that contains all the packages that are utilized for bioinformatics. Okay. So, you will see a lot of these tools for transcriptomic data analysis are present in bioconductor. So, the first one is the dsec2. Okay. So, we will use this dsec2. You can search here on top, you can see this search button on top here, right, and we can simply search dsec2, right, and this will come. Okay. So, the first option you can see bioconductor dsec2, and you can see the package name, right. So, you have differential gene expression analysis based on the negative binomial distribution, right. It shows it gives you the author names, the paper, right, the reference etcetera. Now, the installation of these bioconductor packages is slightly different, you will see, right. First you have to use these commands, it tells you what the commands are. Now, this is under version 4.3, okay, this is the latest version, right. You can simply copy this and type on your R console. Okay. So, I am simply running that, okay, and this is already present in my case bioconductor 3.17, and then you write biopsy manager install dsec2. Okay. So, the first command, this first part of this command is installing biopsy manager, which will manage installation of all the packages from bioconductor in future. Okay. So, once you have installed this, you do not have to install this again. Okay. So, let us say we say dsec2. Okay. We want to install this package dsec2. So, it will take some time and it will tell me okay, that this is already installed because it is, right. So, I have already installed this. So, it should find this installation and it might just say okay, this package is already installed, okay, depending on the internet connection. Okay. Okay. So, I said like no, I do not want anything. Okay. So, maybe it has found some updated version of dsec2. Now, we can load this dsec2, right, and see whether it loads here. Okay. So, it should come here, okay, and in a moment it should load. Okay. Some of the things might take a bit of time because they are quite heavy. Okay. So, you can see it is loading when you type this library dsec2, it is loading other packages that are required for this package. Okay. So, similarly you can see a lot of commands, right, but as long as you are not getting any error, right, you are fine. Okay. We can also try another package, right. So, that we will use is the okay. By the way, I remember these packages that we need. So, I am just installing them, but as we go on, if we see some packages are missing, we can install them one after another. Okay. So, again it will ask for updates. So, I must say no because it might take a long time, and it is saying me here that this package is not installed, right, because this is already present. Okay. The version that I am to install is already present. So, I do not have to install again. So, it is just giving me this message. Okay. And similarly, we can load this, right, library ruvsec, sorry, spelling again you have to be careful about this, right, and it will load this package here. We can use the commands inside this package for all our analysis. Okay. So, to summarize, we have now seen, right, where we can get these data sets, right, from this NCBI geo database, right, and we can try these different sorts of things in R, right. So, that is what I will encourage. If you have your own data set, that is great, then you can actually load in R once you have obtained the count data, and you can load in R, and you can then start doing this all sorts of analysis that we are doing. I have also shown you how to install packages, right. So, any package that you need for your analysis, you can use this install.packages command, and it should be installed very quickly, and you can load these using this library command. Okay. And finally, there are packages that are utilized for bioinformatics, which are available in the bioconductor. Again, the installation command is slightly different, but again you can install this very easily from the R console. So, that is it for this class. So, in the next class we will start with the preliminary analysis of the data, and we will follow the steps one after the another. Thank you very much.